Today we will discuss about the translation part. Okay, so in the previous class we have seen that the central dogma where forming of mRNA from the DNA is known as the transcription, and like that when the mRNA getting converted into the protein or amino acid, it is called as the translation. Okay, so this translation, it this ribosome translate the genetic message of mRNA into the protein. Okay, so we got from the transcription we got the mRNA, and from this mRNA, by using the ribosome and certain enzymes, it will produce the chain of the amino acid or the polypeptide chain, that is called as the protein. Okay, so this mRNA is translated from 5 days to 3 days end. Okay, and this 5 days end of the mRNA, here this is the 5 days end and this is the 3 days end. Okay, so this 5 days end is called as the N terminal. Okay, and this is called as the C terminal, or we can say it is the amino terminal, and this is the carboxy terminal of the this polypeptide chain or the mRNA. Okay, not mRNA, because in mRNA there is a basis. Okay, but in amino acid there is a single amino acid is made up of amine group as well as the carboxy group. So that's why when it is synthesized as a polypeptide chain, that time this 5 days end is known as a N terminal, and this 3 days end is called as a C terminal. Okay, so this amino acid bound to tRNA are inserted in proper sequence. Okay, because of the specific binding of each amino acid to its tRNA. Okay, so there is for every amino acid there is a particular tRNA, but there is 64 codons, but there is no 64 tRNAs. So because of the Bobel hypothesis, one tRNA can charged 2-3 amino acids. Okay, so that's why it is not equal to the number of the tRNA is not equal to the number of codons. Okay, but it is basically depend on the specificity by binding with the tRNA. Okay, and this specific base pairing between the mRNA codon and tRNA anticodon position, the binding, the base pairing, how this tRNA will know that this is my amino acid. So there is in mRNA, in sequence level, suppose this is the UUC, like that in mRNA, if it is the sequence, then tRNA should have the AG sequence. Okay, so this is called as the proper base pairing between the mRNA codon and tRNA anticodon position. Okay, so then only this tRNA will after means matching with the codon, it will know that this is this amino acid I have to charge first. Okay, so based on the base pairing it will know. And where the base pairing is happening, it is happen between the mRNA codon and tRNA anticodon. Okay, so there is a different component which is necessary for the translation. Okay, so the first component is the mRNA. Okay, so in case of the eukaryote, the mRNA is made in the nucleus and transported to the cytoplasm for the protein synthesis. Okay, but in case of the prokaryote, there is no nuclear membrane. Okay, there is only cytoplasm, and so there, that's why the mRNA is synthesized on the cytoplasm, as well as it is converted into the protein in the cytoplasm itself. Okay, so there is, because there is no nucleus, so that's why it is transcription and translation occur concurrently. Okay, so it is going it is the simultaneous process. Suppose this is the mRNA, so from this mRNA, while it is synthesizing the mRNA, it is also producing the polypeptide chain. Okay, so in prokaryotic, there is, because there is the absence of nuclear membrane, that's why the transcription and translation took place together. Okay, but in case of the eukaryote, there is the different compartments, so that's why the the mRNA will produced in the nucleus and then transported to the cytoplasm, where the ribosome will come and bind to the mRNA and produce the protein. Okay, the second component is the tRNA. Okay, so we can say it is act as a adapter molecule. Okay, and that mediate the transfer of information from nucleic acid to protein. Okay, so it is helping in, as the name suggests, tRNA means transfer RNA. Okay, so it will help, firstly it will like charge the amino acid. Okay, then it will transfer this particular amino acid to the particular site of the synthesis. Okay, so here if we and third one is the ribosome. Okay, so ribosome is the major manufacturing unit of the cell, which is located in the cytoplasm and contain the ribosomal RNA and protein. So this ribosome is majorly made up of ribosomal RNA and proteins. Okay, and it is located in the cytoplasm. When this mRNA transferred into the cytoplasm, this ribosome through its subunit, it will bind to the mRNA on a particular site, and then the protein synthesis will occur. Okay, so here you can see that this is the bacterial ribosome and this is the mammalian ribosome. Okay, so there is in both the cases there is two subunits. Okay, in case of the bacterial ribosome there is 50s subunit and 30s subunit, but in case of the mammalian ribosome it is 60s subunit and 40s subunit. Okay, and this 50s subunit is made up of 23s RNA, 5s RNA, and plus 31 proteins. Okay, and this 30 subunit is made up of 16s RNA. Okay, so here this 23s RNA, which is the major subunit of the 50s subunit, it act as a catalysis. Okay, means it will work as a peptidyl transferase. Okay, so it will help to migrate this ribosome from one place to another place in the on over the mRNA. Okay, so it work as a catalyst. Okay, the second the major component of this like in bacterial ribosome is the 16s RNA. Okay, so it is helping in the recognition of means placing the ribosome. Suppose this is the RNA, so where this ribosome will bind in the RNA, that is find out by the 16s RNA. Okay, and likewise this in case of mammalian or in case of eukaryotic there is also two type of subunit, 60s subunit and 40s subunit. So it is made up of 20s rRNA, 5.8 rRNA and 5s rRNA and 49 proteins. Likewise, this 40s subunit is made up of 18s rRNA and 33 proteins. So here also this 28s RNA is working as a peptidyl transferase, and this 18s RNA will work as a recognition site. Okay, and if you will closely look onto the ribosome structure, then you will find out this is the small subunit and this is the large ribosomal subunit. And this large ribosomal subunit, there is three sites. One is the E site, P site and A site. Okay, so this is the arrival site. Here it will transfer the like elongate here the amino acid, and from here the remaining things of the polypeptide chain will exit from here. Okay, so this is the structure, or this is the organization of the ribosomal subunits. Okay, and this is the structure of tRNA. If you will see here, this is the lower portion, there is the anticodon position is there. Okay, and from here this part only, like it is having the complementarity to the mRNA coding coding with the mRNA codons. Okay, it is having the complementarity base pair with the mRNA codons. Okay, and there is one CCA group is there. Okay, here in the tRNA, if you will see here in the 3' position, there is a CCA group is there. Okay, which is helpful to like bind with the amino acid. And here you can see that this amino acid is bind with the this CCA part, okay, of the tRNA. Okay, so this amino acid attached by carboxyl group to ribose of last ribonucleic acid of tRNA chain. Okay, so this is called as a I forgot what's name. This is the like uh, sorry, I forgot its name, what it's called. Okay, this is the site where like the amino acid will come and bind to the tRNA, and from here only it will be charged. Okay, and this is the anticodon site of the tRNA. Okay, so this is the different part of the tRNA, and apart from that there is enzyme required for the attachment of amino acid to the correct tRNA molecule and for peptide bond formation between two amino acid. Okay, so two type of enzyme is required. First one is for the attachment to the tRNA molecule, and second one is the peptide bond formation. Okay, and the proteins, which is nothing but it's also like enzymes, and this some kind of soluble factors also require for the termination of this protein synthesis. Okay, and initiation, elongation also require certain kind of transcription factors, that is nothing but the proteins. Okay, so these are the different component of the translation. Okay, so then as I told you, there is like the number of codon as well as the number of tRNA is not same because of the vowel hypothesis. Okay, so what is the vowel hypothesis? Vowel hypothesis is saying that it is occur in the anticodon position. Okay, so where is the anticodon position? Anticodon position is present in the tRNA. Okay, so this yeah, I remember that this its name is acceptor arm of the tRNA. Okay, which is accepting the amino acid. Okay, on the 3' end of the tRNA. Okay, so that is called as the acceptor arm, and this is called as the anticodon loop. Okay, so this in the this yellow color highlighted region you are seeing here, so that is called as the vowel position. Okay, so in this vowel position what is happening is, because it is the last residue of the codon. Okay, so that's why it is structurally, if you will see, this region is not very much compact or not very much like constrained, so it is kind of flexible, so it can bind with any type of the like amino acid. So that's why it is called as the vowel position. Okay, so the third base in the codon is able to base pair less specifically because it is less constant three dimensionally. Okay, so the binding will not as much strong as these two position, but it can it is little bit flexible so that it can bind with non specifically to other amino acid. Okay, so that's why it is called as the vowel position, and it allows that tRNA anticodon to recognize up to 3 different codons. Okay, so here if you will see, this is the code for phenylalanine, that is the AAG. Okay, so in mRNA it can be either UUU triple U, or it can be UUC. Okay, so this G can base pair either with the U. Here you can see it can base pair with the either U and C. Okay, ideally it should base pair with the C only, because the G is always base pair with the C, so that's why it should base pair with the C, but because it is present on the vowel position, so it can also base pair with the uracil. Okay, and likewise this cytosine can base pair with the guanosine. Okay, and this adenine can base pair with the uracil, and uracil can base pair with adenine and guanine, and this inosine can base pair with the UCA. Okay, this is the only amino acid which can base pair with the three different type of like amino sorry, different type of base pairs. Okay, if it is present in place of anticodon first position, if I is present, then it can base pair with the three different type of codons. Okay, so you have to remember this part, like the vowel position is always present between the anticodon loop of tRNA and codon region of the mRNA, and the third position of the This third position of the codon is called as the vowel position. Okay, and because there is the less constraint, that's why it can base pair with the others base pair also. Okay, so this means this ionosine can only bind with the three different type of base pairs. Okay, apart from that, this G and U can bind with two different type of base pairs. Okay, so that also you can remember. So the mechanism of translation, if we will see, there is three steps. First one is the initiation, second one is the elongation, and third one is the termination. Okay, like in other cases, like in case of replication, in case of transcription, like that, in translation also there is three step: initiation, elongation, and termination. Okay, so in initiation, it is means all the component will set the stage for polypeptide chain synthesis. Okay, all the component will come and assemble there and start the process. Okay, in elongation, it can cause the sequential addition of amino acid to the polypeptide chain in a collinear fashion as determined by the sequence of mRNA. Okay, so after initiation, there will be just adding the amino acids so that the polypeptide chain can grow and on the based on the what is the sequence of mRNA. Okay, so then finally there will be termination, which bring the polypeptide synthesis to a halt. Okay, so initiation, if you will see, the initiation codon is always AUG. Okay, the methionine is always the first codon from where the protein synthesis is started. This AUG is coded the methionine amino acid. Okay, it is toward the 5' end of mRNA molecule that is being translated, and not the first three nucleotide. Okay, it is nothing like from the 5' end it will start from here. Okay, it is nothing like that. Okay, it means in case of the bacteria, what is happening is there is one sequence called as the Sain-Delgano sequence. Okay, so this Sain-Delgano sequence is the starting point where the ribosome will come and bind to this Sain-Delgano sequence, and because of this 16S rRNA, as I told you in the previous slide, there is in the subunit there is 16S rRNA and 23S rRNA in case of bacteria, right? So this 16S rRNA will working as a scanner, okay, on the mRNA. So this will act as a means there is a proper base pairing between this 16S rRNA and the mRNA, and this position is called as the Sain-Delgano sequence. Okay, so from here only the ribosome will after base pairing it will completely placed on this mRNA, and from here after 7 nucleotide there is AUG will fall down, and from here it will start the synthesis of protein. Okay, so this is the mechanism of initiation, like how it is started. So in prokaryotic there is a conserved region about 7 nucleotide upstream of the initiating AUG. Okay, so suppose there is AUG, so upstream of this AUG, 7 nucleotide upstream of this AUG, there is a one sequence that is called as the Sain-Delgano sequence. Okay, so in the small subunit there is 16S rRNA which is having the complementarity towards this Sain-Delgano sequence. So once this complementary base pairing is occurred, then ribosome get to know like that this is the proper place where I have to sit and start the initiation. Okay, so this is from here the ribosome will bind, and after 7 nucleotide there is AUG, the starting codon or the methionine. From there the polypeptide chain synthesis will start. Okay, so this Sain-Delgano sequence is complementarity to a region at the 3' end of 16S rRNA of the small subunit, and base pairing between these complementary sequence stabilize the binding of ribosomal subunit to mRNA for proper assembly. Okay, so because of this base pairing only the ribosome can properly hold the mRNA and start the protein synthesis. In case of the eukaryotes, what is happening is like in prokaryote, the first AUG is recognized by special tRNA, okay, that is called as the formyl methionine tRNA, okay, which carrying the modified methionine, formyl methionine, codes for that methionine, and the large subunit of ribosome now attached to the small subunit to complete the initiation process, okay. And but in case of the small eukaryote, what is happening is there is no kind of such kind of signed Delgado sequence. What is happening is this is the like eukaryotic initiation, okay. So suppose there is AUG, okay. So firstly the small subunit will find out this methylated cap of the 5' end of mRNA, okay. So there is in the eukaryote there is 5' end is methylated, okay, of the mRNA is methylated, so that the 40S subunit of ribosome can determine this particular position, and once it is find out this methylated cap, it will slide on this mRNA and go to this AUG site, okay. And this AUG is part of the Kojak sequence, okay. It is nothing like means if AUG is fall down here, it will accept it as a like initiator codon or a starting codon, okay. The AUG should be part of the Kojak sequence, okay. That is the another rule for the initiation of translation in case of the eukaryote. The first thing is the 40S subunit should find out the methylated cap of the 5' end. The second thing is this AUG should be part of the Kojak sequence, okay. So these two things when happen, and unlike in means case of the prokaryote, there is formyl methionine as a first amino acid, but here there is only methionine, okay. In case of the eukaryote, here you can see this is the mRNA and this is the AUG. So the formyl methionine, this is the initiator tRNA, okay, and it will come and complementarity with this mRNA, okay. So then the 50S ribosome subunit will come and bind with this small subunit, and here it will complete the ternary complex, okay, where the ribosome tRNA and rRNA is present, okay. And there is the several transcription factor which help during this procedure, okay. And this is the energy driven process, so that's why it is like the GTP is required for this process, okay. And likewise in case of the eukaryote, there is 5' cap is methylated cap is required on the mRNA, then only this 40S subunit can come and bind here and scan the Kojak sequence, where the AUG it will find, it will start the protein synthesis from there, okay. And then after reaching to this AUG side, then its bigger subunit, that is the 60th subunit, it will come and bind to the small subunit and form the complete complex, okay, like in case of here, okay. So got the difference, like and it make the 80S initiation complex, here it make the 70S initiation complex, okay. Both are the energy driven process, but in case of the prokaryote, here it is taking GTP and here it is the ATP, okay. And here the first amino acid is the formyl methionine, here only methionine, okay. And here signed Delgado sequence is working as a starting site or the recognition site. Here it is the Kojak sequence, which is the the AUG should be part of this Kojak sequence, okay. So after initiation there is elongation, okay. So at the start of the elongation, the mRNA is bound to the complete 2 subunit ribosome, okay. Here the mRNA is bind with 2 subunit, the bigger subunit and the small subunit, and form the complete complex, okay. So the mRNA is bound to the complete 2 subunit with the initiating tRNA in the P site, okay. So here we have seen there is 3 sites, right. So E site, P site and A site, right. So this P now the tRNA, the first amino acid is present on the P site, okay. So this yeah, this initiating tRNA is present on the P site of the ribosome, and the A site is now free for binding of the next tRNA. Okay, so whatever next tRNA will come, that will come from A site. Okay, here this A site here, and the P side there is the there should be always first amino acid will be there only. Okay, and from A side the new amino acid will come, and here the peptide bond formation will happen. Then this amino acid will go transfer to the P side. Okay, so the ribosome moves along the mRNA in 5' to 3' direction in a stepwise process, recognizing each subsequent codons. Okay, so like if suppose there is UUC, so it will the tRNA will take the phenylalanine, because this is the codon for phenylalanine. Okay, so it will charge the phenylalanine and come from this A site. Okay, then there will be peptidyl transferase enzyme, then which catalyze the formation of a peptide bond between the free N terminal of amino acid at the A site and the carboxy end of the amino acid at the P site, which is actually connected to the tRNA. Okay, like if you will see here, yeah, this is present as the P site. Okay, and this is the new amino acid which is present as the A site. Okay, so what peptidyl transferase, what it will do is it will create the peptide bond between these two amino acid, the initiated amino acid and the next amino acid. Okay, whatever it is, so it will, what it will do is it will connect the carboxy terminal of the initiator amino acid to the amino terminal of the next amino acid. Okay, so this and it is being present as a P site, the starting codon, and this next codon is present as a site. Okay, after peptide bond formation, the what will happen is this ribosome will little bit move from P site to A site, and this new peptide bond now formed, so this amino acid will come to the P site. Okay, so this disconnect the tRNA F met from the amino acid, and tRNA at the A site now carry two amino acid. Okay, because sorry, this it will be present in the A site, not in the P site. Okay, after like formation of the first peptide bond, this two amino acid will move to the A side, because ribosome is moving from P to A side, right? So that's why this is moved to the A side. There is two amino acid now is there. Okay, with a, and now the tRNA F met will remove from this E site. Okay, so this with a free N terminal, carboxy terminal of the second amino acid now connected to its tRNA. Okay, so this is now N terminal and C terminal of the second amino acid is still connected to the tRNA. Okay, when the third amino acid will come, that time this second tRNA will be also removed from the E site. Okay, understood? So what is happening in the starting, in at the time of initiation, what will happen is the tRNA will come and bind to the P site. Okay, so in case of bacteria it is F methionine, and in case of the eukaryotic it is methionine only. Okay, so once the next amino acid will come from this A site. Okay, so this as I told you, this bigger subunit is having the 23S subunit, and in case of eukaryotic it is having the 28S subunit. Okay, it is having the it is act as a catalyst. Okay, which kind of catalysis it is doing? It is doing the peptide bond formation. Okay, so peptide bond formation, if A side the amino acid is coming, it will create the peptide bond between these two amino acid. Okay, so after once this peptide bond is formed, this amino acid is transferred to the A side, and this tRNA is now free, so which is removed from this complex through the E side. Okay, now this second amino acid is still attached to the like its tRNA. Okay, so once the third amino acid will come, that time this second tRNA will also be like free from this step, and it will exist from this complex through the E site. Okay, so like that this process will continue, and this process is called as the elongation, because here the chain is now growing. Okay, so the last step is the termination. Okay, so termination, when the ribosome encounter a stop codon, there is no tRNA available to bind to the A site of the ribosome, right? So this is the phase where it find out that the protein should be like here the peptide bond should be released from here. Okay, the details are not very clear, but once the release factor binds, the ribosome unit falls apart. Okay, so there is certain kind of the transcription factors, when it bind to this complex, then this ribosome assembly will disturbed, and the protein synthesis will halt there. Okay, so the tRNA carrying the polypeptide is also released, freeing up the polypeptide product. Okay, so there is different kind of the releasing factor. Okay, so when this will someone will release the mRNA, someone will release this tRNA, and someone will release the protein. Okay, the polypeptide chain, and this ribosome will be dissociate from the mRNA. Okay, so because the tRNA which is coming from like A side, there is no tRNA, means charged tRNA having the amino acid, so then this complex will got to know that we have to stop the synthesis. Okay, so then releasing factor will come and bind here, and it will release each component from this assembly. Okay, so this is the complete process of the like protein synthesis. Okay, so firstly what we have seen is the initiation started in case of the prokaryote. It is the in 16s RNA of ribosome. It is having the complementarity base pairing with the signed-Delgado sequence present on the mRNA. Okay, after proper base pairing, the ribosome will come and sit here. Okay, and once it is proper binded with the mRNA, it start the synthesis of protein downstream of 7 nucleotide of this signed-Delgado sequence. Okay, and there is AUG codon. Okay, after 7 base pair there is AUG codon which will code the formyl methionine. Okay, so this from here the protein synthesis will start. Okay, but in case of the eukaryote, what is happening, there is nothing like signed-Delgado sequence. There is the mRNA is already methylated at the 5 days end, that is called as the methylated cap. Okay, so this 40s subunit, through the help of 18s rRNA, it will recognize this methylated cap, and after recognizing, it will slide on this mRNA and reach to this AUG site which is present on the Kojak sequence. Okay, so this after reaching there, it will start the, from here it will start the protein synthesis. Okay, means after reaching here the small subunit, the large subunit come, that is the 60s subunit, and it will create the proper assembly of the ribosome, and from here the protein synthesis will start. Okay, and here the first amino acid will with tRNA, it will come from the P site of the ribosome. Okay, then in the next step, in the elongation step, what is happening is now the larger subunit is also there. Okay, so through the help of 23s RNA or the 28s RNA, what it will do is means the upcoming amino acid, it will create the peptide bond formation between the amino acid which is present in the P site and amino acid which is coming from the A site. Okay, here it will create the peptide bond formation, and now after peptide bond formation, what is happen is this first tRNA will come to the E site. Okay, and come to the E site, and this P site is now free, and this A site is having 2 amino acid. Okay, the second amino acid is now bind with the its tRNA, which is already means still attached to this amino acid. Okay, then again when next amino acid will come, this tRNA will shift to this p side, and again this larger subunit with the help of 23-20S rRNA, it will create the peptide bond formation, and it will shifted to the A side. Okay, so like that it will means the older tRNA will remove from this E side, and the upcoming amino acid is coming from this ribosome through the A side. Okay, and this whole process is means driven by the help of energy. That is, in case of prokaryote it is the GTP, and in case of eukaryote it is the ATP. Okay, so this is the process of elongation, and there is also termination, because there is no for stop codon there is no tRNA. Okay, so that's why this A side will be like blank. Okay, so that's why from here this complex will get to know like that here we have to stop the protein synthesis, because there is no tRNA base pairing. Okay, so the certain releasing factor will come and bind here, which will release this tRNA as well as this mRNA and this polypeptide chain, and after that this ribosome subunit is also disassembled. Okay, so this is the termination process. So this is the certain difference between the prokaryote and eukaryote gene expression. If we will see, there is no nucleus in case of the prokaryote, so that's why the transcription and translation take place in the same cellular component. Okay, and the translation is often coupled to the transcription. Okay, so here in case of the prokaryote, there is only the cell membrane is present, there is no nuclear membrane, so that's why the transcription and translation, both the thing is happened together. Okay, and in the same place. But in case of eukaryote, there is the nuclear membrane as well as the cellular membrane, so that's why in nucleus there is the transcription occur, and in cytoplasm, after transferring the RNA into the cytoplasm, there is the translation. Okay, and second thing is gene, here in case of prokaryote the genome is very small, so that's why it is not like divided into exons or introns. Okay, but in case of eukaryote, because the gene is very much complex, okay, so it is the DNA of a gene consists of exons separated by the introns, and exons are defined by the post transcriptional splicing, which delete the intron. Okay, so there is after transcription what is happen, there is certain kind of means processing will happen, where this intronic part will be removed and only the exonic part will be there, which will code to the any kind of gene. Okay, but it is absent in case of the prokaryote. It is like a straightforward, whatever sequence is there, that code for the protein, but here the intronic part is the waste part of the gene. Okay, so it will be removed by the further processing. Okay, so the in transcription level, one RNA polymerase consisting of 5 subunit. Okay, so the primary transcript are the actual mRNA, they have a triphosphate start at the 5 days end and no tail at the 3 days end. Okay, so like in case of transcription, because it is very straightforward, means the gene is not containing any kind of the intron or exon, it is only exonic part, so that's why there is no requirement of splicing. Only this 5 days 3 days, means in the previous class we have seen that the 5 days end is only having the triphosphate, remaining base pair is having only single phosphate group in between. Okay, so that's why this there is no processing required. This triphosphate itself is as a recognition site, it will work as a. Okay, and in case of this eukaryote, several kind of RNA polymerase is there, and each containing 10 or more subunit, and different polymerase transcribe different genes. Okay, So unlike this prokaryote, there is different type of RNA polymerase, okay, which is having different kind of like subunit, and different polymerase will transcribe different RNA. Okay, in the previous class that we have seen that single RNA polymerase in case of the prokaryote, it can make all kind of RNA like 15s, 16s, 18s, like that it can make. But in case of eukaryote, for making different type of RNA, there is different type of RNA polymerase. Okay, three different type of RNA polymerase present in the eukaryote, 1, 2 and 3. Okay, and this primary transcript undergo processing because it is having the intronic and exonic part, so that's why it is undergo the splicing process, okay, to produce the mature mRNA which is having only the exonic part, and that have a methylated cap at the 5 days end and a poly A tail at the 3 days end. Okay, so like in case of here, because here signed Delgado sequence is not present, so that's why the methylated cap is required to protect this mRNA from when it is transferring from nucleus to cytoplasm, there is so much chances of RNA activity and it can degrade this mRNA. So that's why to protect this mRNA, there is methylation happen in the 5 days inside. Okay, so that's why it is 5 days methylation happen, and from this methylation site it is also act as a recognition site for the translation. Okay, so once it is methylated in the during the splicing and all, there is also formation of poly A tail, okay, which will also helpful in case of translation. Okay, so to bind with the transcription factor it is required. Okay, so in case of translation, there is unique initiator tRNA carries the formyl methionine, which is absent in case of the eukaryote. It is only methionine. Okay, and here the mRNA have multiple ribosome binding site and can thus direct the synthesis of several different polypeptides. Okay, it is polycistronic because the transcription and translation both occur at the same time, right? So that's why from each means single mRNA there is different ribosomal binding site, different in starting codon, okay, so which can code different gene from the same mRNA. Okay, but in case of this eukaryote, there is a different location, the transcription and translation location is different, so that's why mRNA have only one start site and can direct the synthesis of only one kind of the polypeptide chain. Okay, and in case of here prokaryote, there is small ribosome subunit immediately bind to the mRNA ribosome binding site, and in case of eukaryote, there is a small ribosomal subunit bind first to the methylated cap at the 5 days end of the mature mRNA, and then scan the mRNA to find the ribosome binding site. Understood? So here because it is in case if you will see the overall process, the prokaryote is not having the nucleus, so that's why both the process occur simultaneously. But in case of eukaryote, there is proper chamber for occurring each event, so that's why transcription take place in the nucleus and translation take place in the cytoplasm. Okay, so but and the second thing is its gene is only having the exonic part or only having the coding region in case of prokaryote, but in case of eukaryote there is non essential part is also there, that is called as the intronic region, which doesn't code any kind of the gene, so that has to be removed. Okay, in the transcription level there is single RNA polymerase, okay, which synthesize all kind of the RNA, and in case of eukaryote there is different type of RNA polymerase. There is basically three type of the RNA polymerase which synthesize different type of the RNAs. Okay, and once this RNA different type of RNA is made, because there is intronic and exonic part, so this RNA will go for the further maturation. How it will mature? It will splice the intronic part, and then because it has to transport from the nucleus to cytoplasm, that's why it has to cap cap, methylated cap at the 5' end, and should also have the poly 8L at the 3' end, which will require for the binding of certain transcription factor for the translation. Okay, so these two things also happen during the maturation process of the mRNA in case of the eukaryote. And in prokaryote, that is very straightforward, nothing is required, just 3' triphosphate group which is present at the 5' end is means enough for the start of the synthesis of translation. Okay, because here the first thing in the translation level, if you will see here, the formyl methionine is coming in case of prokaryote, but here it is the normal methionine. Okay, and the mRNA have the multiple ribosome binding site because it is polycistronic, so that's why each means only single mRNA can code different type of the gene. Okay, and different type of the gene or different type of the polypeptide chain, but here the event occur in the different location, that's why the mRNA in case of the eukaryote is monocistronic, and it is coding only single type of the gene. Okay, or the polypeptide chain, and here because there is nothing required, so that's why the small ribosome can go and directly bind to the Sine-Delgado sequence on the prokaryote. And in case of eukaryote, there is firstly the methylated cap will be scanned by the small subunit, and then after means sliding onto the AUG site, it will start the protein synthesis from here. Okay, so these are the difference between the prokaryote and the eukaryote gene expression. Okay, and there is a different inhibitor of protein synthesis. Okay, like in case of transcription and DNA replication, here also there are different inhibitor which we can use as a drug target. Okay, so if you will see, protein synthesis is a target of a wide variety of naturally occurring antibiotic and toxins, and mechanism of action of some common antibiotic and toxin which inhibit protein synthesis in prokaryotes and eukaryote are described. Okay, so first one is the streptomycin, which is most common antibiotic. Okay, and it is the basic trisaccharide. Okay, how it will inhibit the protein synthesis? By binding with the 30th subunit. That you have to remember, like how it is acting, how it is inhibiting the protein synthesis. So streptomycin is bind with the 30th subunit of the small ribosome. Okay, of the bacterial ribosome. Okay, because from 30s you can understand like it is bacterial ribosome, because in eukaryote there is 40s ribosome subunit, right? So that's why it is bind with the 30th subunit, small subunit of the bacterial ribosome, and cause the misleading of mRNA at relatively low concentration. Okay, so how it means inhibiting it, it will in the small subunit when it will bind, then it will hamper the activity of 16 sRNA, which is helpful for reading the proper location where have to the ribosome has to sit, right? So it will bind with the 30th subunit, and it will like hamper the identification of proper location. Then second antibiotic is the chloramphenicol. Okay, so chloramphenicol bind to the 50S subunit and block the peptide bond formation through the inhibition of peptidyl transferase, but doesn't affect the cytosolic protein synthesis in eukaryotes. Okay, so what it is saying is chloramphenicol bind to the 50th subunit. Okay, and we know that 50th subunit is containing the 26 sRNA. Okay, and but it is this 50th subunit when it is binding, it means we are talking about the bacterial ribosome, not the eukaryotic ribosome, right? So it hampers the 26 RNA activity, because this 26 RNA is performing the peptidyl transferase activity for peptide bond formation, so it will block this peptidyl transferase activity. But in case of, if you will give this chloramphenicol to eukaryote where the protein synthesis happen in the cytosolic part, it will not inhibit the protein synthesis, right? Because here the subunit is also different. Here there is 60S subunit, but in case of eukaryote, in prokaryote, there is 50S subunit where the chloramphenicol will go and bind to this 50S subunit, and the peptide transferase activity will get lost. Okay, And third one is the tetracycline. So this tetracycline is again bind with the 30th subunit and interfere with the amino acid tRNA binding. Okay. So this is means in the P side there it will inhibit the binding of this amino acid tRNA. Okay. So this is also like work on the prokaryotic ribosome. Okay. Then erythromycin, it will bind to the 50s subunit and inhibit the peptide chain elongation. Okay. It terminate the elongation part. Okay. And then eukaryotic inhibitor, if you will see, there is cyclohexamide which block the peptidal transferase of 80s ribosome but not the 70s bacterial ribosome. Okay. In if you will give the cyclohexamide, it can only inhibit the eukaryotic ribosome, not the prokaryotic ribosome. Okay. And there is diphtheria toxin. It is exotoxin of Corynebacterium diphtheria infected with a specific bacteria. Okay. It stop the protein synthesis in eukaryote by inactivating the elongation factor 2. Okay. So by binding with this elongation factor 2, it reduce the availability of this elongation factor 2. So that's why the protein synthesis will be hampered in case of the Corynebacteria. Okay. The third one is the ricin A. Ricin A is a toxic protein of castor bean which inactivate the 60S subunit of eukaryotic ribosome by depurinating a specific adenosine in 28S rRNA. Okay, so it will modify the adenosine which is present in the 28S rRNA. Okay, so that's why it can't it can't do its proper activity because 28 srRNA is having the peptidal transferase activity, so it won't perform its activity. Okay, so this is the different type of inhibitor. You have to remember each of the inhibitor how it's functioning. Okay, where it is going and bind to what. Okay, so has been asked for their efficient translation, eubacterial mRNA possesses a Sine-Delgado sequence for its recognition by anti-Sine-Delgado sequence in the ribosome. The correct statement is this anti-Sine-Delgado sequence is present in the 5S rRNA, present in 23S rRNA or 16S rRNA, or it is formed by the interaction of 16S rRNA with 23S rRNA upon docking of the 50S subunit on the 30S subunit of the ribosome. Yes, the correct answer of this question will be 16S. This as we have seen here. Here the 16S rRNA which is present in the small subunit of the ribosome. Okay, this is called as the anti-Sine-Delgado sequence because Sine-Delgado sequence present on the mRNA, so anti-Sine-Delgado is present on the 16S rRNA which is the part of the small subunit of the ribosome. Okay, so that's why it is not made up of the docking of 50S or 30S. Okay, it is only part of the 30S. Okay, 30S 16S rRNA of 30S. Okay, so that's why the correct answer of this question will be 16S rRNA. Okay, not this one.